Yeah, sorry guys. Thank you very much for that lovely introduction and such a great background. It really makes my work very easy. So, like it was stated, the topic is elucidating mechanisms of cluster N mycobacteriophage virus prophage-mediated defense. This may be a mouthful, but it essentially means that I am interested in uncovering how virus prophage defends its host, MSMEG, against attack by other phages. And in this talk, I would like to give a brief introduction. I understand I have a very broad audience, so this will definitely come in handy. And then I would like to tell you why we should care generally about phages and specifically about the work that I'm doing. Then I'll show you some results of my work, and you should understand that in 20 minutes or so, it's going to be just a fraction of the numerous data that I have. And in conclusion, I would like to tie all the data points into a model that I've come out with. Hopefully there will be time for questions. So whatever is not clear, hopefully I'll have that opportunity to flesh out some more details. So by way of introductions, when we say bacteriophages or phages, these are viruses that infect bacteria. These viruses do not infect animal cells. They cannot infect plant cells. They cannot infect fungal cells. They only specifically infect bacterial cells. Now we can see these under the electron microscope, and I'm showing you how they appear, some of them. This is the morphology under the electron microscope. And on the right here, I'm just showing you a cartoon of a bacteriophage with the head that harbors the genetic material, in this case DNA, it could be RNA. It has a tail here, and you can see tail fibers, which helps in absorbing to the cell membrane of a bacteria. And one fun fact is that bacteriophages are the most abundant biological agents in the universe. So by way of life cycle, they come in two flavors. So when a phage infects a bacterium, it most likely will hijack the bacteria's molecular machinery, duplicate its genome, and make more copies of itself. And it will eventually lyse the bacterium and go on to infect others. So the viral particles that are produced can go on to infect new bacteria. Now, some phages know only this life cycle, which we call the lytic cycle. These phages are called virulent phages. On the contrary, there are some phages we term temperate phages. These, in addition to the lytic cycle, have an alternate life cycle, which we call the lysogenic cycle. So here, the phage will come in, its genome will be injected into the bacterium, but instead of duplicating the genome and making viral particles, it is going to attach its genome to that of the bacterial chromosome, and it is content to divide linearly as the bacteria divides. Now, when the phage attaches its genome to that of the bacteria chromosome, it is referred to as a prophage. Now, one thing that I have to say is that in this lysogenic cycle, conditions may arise that will necessitate that the prophage will be induced into the lytic cycle, where it will make more copies of itself and lyse the host bacteria. So why should we care about phages at all? So we currently living in an era where most pathogenic bacteria have grown resistance to antibiotics that we have. And people have seen the promise of phage therapy in counteracting this problem. So I'm going to use this example here, this story, to illustrate both points. So sometime last year, we have this paper that came out from this group at the University of Pittsburgh. And essentially, this little girl here, who was 15 at the time, was suffering from disseminated drug-resistant Mycobacterium abscessus infection. This is Isabel's mom, and this is Isabel's doctor here on the far left. Now, Isabel, as you can see on the right here, was given all these antibiotics. Now, none of them was able to help her resolve the infection. Like any mom would do, the mom asked the doctor to do whatever it was that can make the daughter survive this infection. She was actually discharged to go home and die. So Isabel's doctor got in contact with Dr. Graham Hartford here, who is the leader of the C-phages program that Penny alluded to. Now, all the phages that are uncovered in the C-phages program are sent to Pittsburgh for archiving. So Dr. Hartford looked through the archive and found 3 phages that could kill this Mycobacterium abscessus infection. They made preparations of this, sent to England where Isabel was living, and it was administered and she got better. The infection was resolved, and today she's well and living a normal life. Now, this story demonstrates 2 points: that antibiotic resistance is present with us, and number 2, in phage therapy, we have an alternative that can combat this problem. So, but then there are challenges that comes with phage therapy. So, number 1, phage infections are highly specific. What this means is that for every pathogenic bacteria that you want to arrest with a phage therapy, you need to go and isolate specific phages that can infect them. There is a problem because the act of isolating these phages are not trivial at all. Some of them are really very difficult to come by. The second problem is that bacteria will always evolve resistance to phage infections, and so scientists must think about these two. The third problem, which is my area of interest, is the phenomenon called prophage-mediated defense, and I'm going to speak more on this in my next slide. So prophage-mediated defense occurs when a bacteria which houses a prophage defends against other phages. So what happens is the prophage, as you can see here, this bacteria genome is just a cartoon, has some prophages here, and these prophages express certain genes that defend the bacteria from infection or from attack by other viruses. Now, if we want to use phages to kill pathogenic bacteria, then we don't want any prophages to be there mounting defense against the phages that we are employing in the therapy. And so the challenge is that most bacterial genomes actually carry prophages. And to be able to overcome this problem, there is the need for us to understand the mechanism through which these defenses are established. And that is the goal of my research, to understand how Barrett's prophage defends its host, Mycobacterium smegmatis, against heterotypic phages, and I'm considering purple haze and island 3. So I just want to point out to you that Mycobacterium smegmatis here is a soil microorganism, but it is related at the genus level to some very notorious pathogens. Number 1, Mycobacterium tuberculosis, notoriously known for TB or tuberculosis. The second one here, we have Mycobacterium leprae, that is responsible for leprosy or Hansen's disease. Then we have Mycobacterium abscessus, which give rise to several respiratory tract infections, and we encountered that when we talked about Isabel. So the primary question that I have to answer is, how does Barrett's defend against some other heterotypic phages? But to show you my data, I would like to show you how I go about doing my experiments so that you can follow it well. So here I am showing you a Petri dish with agar media on it. Now, agar media essentially has food that bacteria can feed on and grow. So when I take this plate and I spread bacteria on it, the bacteria has food source, so they grow to fill up the entire plate. So notice the change in coloration. So this plate on the right here has bacteria on it. Now, you can imagine that with bacteria, if we put phages on it, the phages will eat up or kill the bacteria. So what is going to happen is that they are going to make a clearing in the area that you spotted. And I've illustrated that here. So you can see here is a bacterial lawn. And from this point going, I have spotted different concentrations of phages on it. And you can see they are eating up the bacteria there and therefore clearing the bacteria at the region where it was spotted. Now you can also notice that going from left to right, the area of clearing appears to be decreasing. It is decreasing because I have done a serial dilution of my phage, and as you go from left to right, the concentration actually reduces. With this background, there are a couple of things that we can do. So for example, we can have two different lawns and ask the question, does a given phage infect both bacterial lawns equally or to the same extent? So in my research, that's exactly what I did to uncover the phenomenon. So on the left here, we have MC²155. This is just a representation for the type of MSMEG that I'm using. So it is a bacteria with no prophage inside. Now, over here, I have another lawn where I have the baddest lysogen. So essentially, this is an MSMEG carrying the baddest prophage within it. Okay. Now, notice here that I spotted purple haze, which is a phage, island 3, another type of phage, happening here. And I spotted different concentrations of that, reducing from left going to right. You can see that they are able to eat up the bacteria all the way to this point, all of them. Notice that when virus is present in the bacteria, purple haze and island 3 cannot infect to the same extent that they did here when virus was absent. So one of the points we can make is that the presence of virus within the lysogen, right, is able to block or defend or abolish the attack that purple haze and island 3 is mounting. So baddest prophage defense against purple haze and island 3. Eponin, you can see there is no effect on eponin. It can infect efficiently. We can do some math. For example, compare the extent of clearing on this lawn and compare it to the extent of clearing here. The ratio is what gives us the efficiency of plating the EOP. When the efficiency of plating is 1, like if you compare for eponin, then it means there is no defense. But when you compare the extent here to the extent here, you will get a value less than 1. Understand? So for example, 1, 2, 3 over 8 is less than 1, and that shows that is defense. But these are in terms of orders of magnitude. So again, bringing you back to the central question is, I want to understand how virus prophage do this. And so what are the factors? What are the genes involved? So to do that, I have to turn to the virus genome. So you can get DNA of virus, you can isolate that, you can get a sequence, and using bioinformatics and wet lab studies like RNA-seq, we can determine where in the genome has genes that are expressed. So over here, you have genes in rectangular boxes that are aligned to the genomic ruler. So the ruler here represents virus genome. So each part has a gene that can be found there. And in all, we have 66 genes, if you count all these boxes. So I have 66 candidate genes to look and ask which ones are responsible for the defense. Thankfully, I didn't have to do that because in 2017, some group did work, which I have the reference here, you can look it up. And they found out that in the baddest lysogen, only a handful of genes are expressed. And these genes are here in the box region. There are 9 of them. So all the others are not expressed, they are switched off. So that means from 66, I just have to consider these 9 genes. Now this is up close with the 9 genes I talked about. Of this 9, we know something about 38. A lot of work has been done on that, and it is called the immunity repressor. Its function is to switch off or repress the expression of all the other genes. So it is the reason why all the other genes are off. We know that. Then there is this gene 37 we call the integrase, we know what it also does. It is responsible for integrating or attaching or stitching the prophage genome into the bacterial chromosome. So we know what that does. What we don't know is the rest of these genes here, 7 in number, which are also expressed in the lysogen. So this therefore became the focus of my experiment. Now, I like to present some 2 lines of reasoning, which is very popular in science. So let's do this thought experiment. So we can think of it this way. Let's say gene 30 here is the one responsible, right? If we assume, say, gene 30 is the one responsible for defense, or it is required, it is contributing in some way to defense, then when we take 30 out, if we delete 30 or knock it out of the prophage, we don't expect to see defense, because if 30 is responsible and it is no longer present, we should abolish defense. We can do that same reasoning for all the others. Now, let's say we do that and we find that 30 is really required. This is necessary. We can also ask, is it doing it alone, or it is doing it in synergy or in concert with some other factors, right? And that is the sufficiency test. So what we do is we can take 30 alone and put it in the MSMEG and ask the question, when 30 alone is present, do we see difference? And these are exactly what I did going forward. So we've already seen the top panel, which essentially makes the point that Bares, when it is present, defends the host bacteria against infection by island 3 and purple haze. We've seen that already. So here on the lower left panel, I have MSMEG with Bares inside, but this time with gene 30 deleted, right? So I have gene 30 deleted, and I'm asking the question, what happens? Now notice that when you delete gene 30, all of a sudden, purple haze, which over here was not able to infect all the way, is now able to infect all the way to this point, right? So that means that gene 30, the absence of gene 30, is allowing purple haze to be able to infect, right? So notice also that for island 3, even as we've taken 30 out, island 3 still cannot infect, just as it couldn't infect here. So that means that there are some other genes that are responsible for defending against island 3. And so we can make the point that Bares prophage encodes at least 2 different defense mechanisms, one specific for purple haze, and that system is different from the system that blocks infection by island 3. The next thing is the sufficiency test that I talked about. We asked the question, now we know 30 is required to defend against purple haze. 30 is necessary to defend against purple haze. So we asked the question, can it do it alone? Is it on its own sufficient to defend? And so we take 30 alone and put it in MSMEG and express it and do the test again and see what we find is that purple haze cannot infect when 30 alone is present. That means 30 alone is sufficient to bring about defense. And just to note also that island 3 is not affected by 30 whatsoever, to make the point. So because of our time, I cannot bring you all the images of all the experiments that I've done, but I can summarize a few things that I've done as well. So we did the same thing for gene 35 and 36. Essentially, I took out gene 35, knocked it out, and I asked the question, is 35 playing any role? Is it required for defense? I found out that it's not required. I did the same for 36 and found out that 36 is also not required. Now, we in our paper, which I think we've circulated to members of the club, and the reference is here, so you can look it up. We were able to show also that GP30, that's Bares GP30, is a cytoplasmic protein. We were also able to show that GP31 is a membrane protein. Now, the way you do this is you have a lot of bioinformatic programs that can predict these things, and you can also just tag the proteins and use microscopy to see where do you find it. Do you find it in the cytoplasm or do you find it on the membrane? And we did these experiments and we were able to also conclude that 30 and 31 interact. So when we do a pull down assay or co-immunoprecipitation, you are able to, when you pull down one of them, you are able to recover the other as well. And I'm not showing data for those. So putting all these things together, we can hypothesize a model through which Barrett's prophage defends against purple haze, for example. So in this cartoon, I'm showing purple haze here as this green phage, and this is the bacterial membrane, which is separating the outside of the bacterium from the inside of the bacteria. What we have here is Barrett's GP31, that is gene product 31, and Barrett's gene product 30. And I'm showing here that they are interacting because we have data that shows that they are interacting. We also have data that shows that one of the minor tails of purple haze will interact with this complex, GP31 and 30 complex. When purple haze docks onto the membrane and this interaction happens, what results is that 30 dissociates. So purple haze docks, interacts with this system, and that interaction causes 30 to dissociate from 31. And when it does that, it comes into the cytoplasm where it is able to block the DNA of purple haze from successfully going through the lytic infection. I must say that we don't know at what specific stage that this arrest happens, i.e. we don't know whether genes are duplicated, whether any genes are expressed, whether the DNA is duplicated, or whether it is assembly of viral particles that is blocked. We don't know. But what we know is that there is an interaction here which will lead 30 to come and antagonize this. Now notice also that we have other these proteins that I have not spoken about, 33, 32, 34, they are all around. And I hypothesize that these guys may in fact be the factors that are responsible for defending against island 3. So, of course, the work is not done yet. We want to continue with deletion analysis. We want to delete gene 31, delete gene 32, 33, and 34, and ask the same question. What happens if they are not present? Do they affect defense against either of the phages that I was speaking about? And because I know the composition, have an idea about the composition of this research club, I'll seize this opportunity to make a pitch that in my lab headed by Dr. Vasi, where we have a lot of research opportunities available, where individual students, typically in their sophomore years, juniors and seniors, can undertake individual research, some related to the work that I'm doing. And of course, we have other interesting questions that are available that you can always take. And of course, for people who want to do grad work, that's grad school, and for other institutions that may want to collaborate, you are welcome to shoot my supervisor, Dr. Vasiwe, an email so that we can have that discussion. So before I take your questions, I would like to seize the opportunity and thank a few people who've been very helpful in this work. So my supervisor, obviously, has been very helpful in this project. I also have a thesis advisory committee who review my work and guide me in this journey. And then I have collaborators that I've been working with. So Dr. Catherine McGinney was a former colleague here who also did a significant part of the work, which we just published. Neda Kutkevich was an undergrad here who took opportunity of the research opportunities in our lab and contributed some data to our recent publication. So she is also a co-author on that. Jamie also did some work in our lab, and her data is not part of this paper. Hopefully in my next paper, her data will be present and she will be a co-author. Now, the staff at the biology department, Lehigh University, have been very helpful in so many ways. And I also like to acknowledge funding sources that has helped me throughout. The biology department of Lehigh University have supported my work through TA and RA grants. I've also received summer fellowship from the College of Arts and Sciences, Lehigh University, and I'm also a recipient of the Lehigh Presidential Fellowship. So I'm very grateful for your attention, and I'm happy to take your questions.
I have a question. So when you manipulate the gene expression, like with like the different, how do you like turn them on and off? Like by what methods do you do that?
Okay, so yeah, that works. So when let me bring this. Okay, so here, so the Barres over here we have the entire Barres here. The entire Barres phage has its own promoters that drive the expression of various genes. Now over here too, the same system is working, but for this one, which is what is represented here, for example, what I do is I clone the entire gene with its promoters. So within the genome, we have promoters that drive the expression of certain genes. So I try to clone the gene with its promoters so that it will be the same native promoter that is driving the expression of these genes. So in this case, I'm just using promoters that are native to the phage, and I'm not using exogenous promoters at all. Yeah.
Thank you.
I don't know. Yeah. Thank you for the question.